Hello everyone. My topic is about heterospory and the origin of seed habit. Heterospory. It is a phenomenon of production of two different types of spores, which is microspore and megaspores, which are they varying in size, structure, and function, and formed in the same plants. They are formed in a sporangia. The microspores are smaller than the microsporangia, and the larger one is a megaspore, which is formed in a megasporangia, respectively. Differences in the size of the spores is related to the sets of the gametophyte. Microspores give rise to the male gametophyte, where the megaspores give rise to the female gametophyte. The differences between the microspores and megaspores is the size. Microspores which are smaller in size, while the megaspore is larger in size, and microspores produce antheridia, while the megaspore produce archegonia. The flagellated are motile and they can swim in the water. They are non-motile. Megaspores are non-motile. So this is a megaspore and microspore within the megasporangia and microsporangia, which has been seen in the Selaginella. They are commonly found in 9 genera of pteridophytes, that is, they are Selaginella, Isoids, Stylites, Marsilia, Pilollaria, Reglinides, Salvinia, Azola, and Platyzoa. These are the 9 species which has been found in a heterosporic life cycle in pteridophytes. These are the heterosporic life cycle in pteridophytes, in which a sporophyte which gave rise to microphyll and a megasporophyll, which is going to microsporangium and megasporangium. After doing meiosis, it converted into microspore and megaspore, and microspore developed into male gametophyte, where the megaspore developed into female gametophyte. From the male gametophyte, antheridia is developed, and from the female gametophyte, archegonia is developed. The anthrozoids and the egg fuse together to form a zygote, which is formed from the antheridia and archegonia respectively. The zygote will be a diploid one, and from the zygote is developed into an embryo to a young sporophyte, and young sporophyte to the sporophyte. This is haplodiplontic life cycle, where both haploid and the diploid phases will occur. An origin of heterospory is due to the reduction in number of spores within the sporangia. When the nutrition become a limited factor for it, the spore functional is competition for the nutrition, and there will be limit for the food supply in microspore. Where in megaspores, the mother cell disintegrate, where we will get the sufficient nutrition for development, and it will be in a larger size. There are 3 evidence for the heterosporic origin of heterospory, and they are paleobotanical evidence. This is by Scott in 1894 in 2 species of Calamostitis, with Calamostitis biniana and Calamostitis cashiana. In Calamostitis biniana, most of the sporangia were within the large number of small spores in tetrads, but in some sporangia, spores were larger. In cashiana, 2 different types of sporangium, some aborted spores in megasporangium, indicating that megaspores were formed at the cost of abortion of the sum of the spores in the sporangium. The other examples are the species of Lepidocarpon, Calocarpon, and Stautoterpis. And the second evidence is developmental evidence. In developmental evidence of heterospory, there are many different stages. First is formation of sporocytes, meiosis, and maturation of spores. For example, Selaginella is developed in a different, similar to the differentiation of sporocytes. When the microsporangium inside the Selaginella, all the sporophytes undergo meiosis, a large number of microspores formed. While megasporangium, all the sporophyte abort except one survive sporophyte undergo meiosis, forming 4 large functional megaspores. 4 large functional megaspores, 3 will be disintegrated and only 1 act as a functional. In Marsilia, both the sporangia are identical until meiosis. This can be attained by the microsporangium, while all the 34s will undergo reduction, where only 1 will undergo the reduction in megasporangium. And the developmental study shows in the determining process of heterospory become operative either before meiosis in Selaginella or Isoids, or after meiosis in Marsilia, Salvinia, and Azola. And the third is experimental evidence of heterospory. This is by Global 1905 in Selaginella and Shartak 1910 in Marsilia. Experiment showed that the heterospory originated due to the nutritional factor. That is, when the low light intensity give less photosynthetic activity, nutrition will become a limited factor, so small size spores will be originate and develop only microsporangia. It is in Selaginella and Marsilia. These are the three evidence for the heterospory. And importance of the heterospores are express sex determination capability of plants. Sets of the gametophyte can be protected at the sporophyte stage. Microspore formed male gametophyte and megaspore formed female gametophyte. The endosporic gametophytic development, so that the nutrition for the developing gametophyte is derived from the sporophyte and not affected by the ecological factor. Ensure nutrition for developing embryo become a megaspore is retained by the parent even after fertilization inside the female gametophyte. And next is seed habit. It is an heterospory condition in which embryo and the rest gametophytic tissues from seeds which germinate to form a new plant. The requirements for the formation of seeds is formation of two type of spores, which is heterospory, and reduction of functional megaspore, and retention of the megaspores in the megasporangium until embryo development, elaboration of upper part of a sporangium to receive microspore polypile available for the sufficient nutrients for the embryo development. These are the steps involved in the seed germination, where the homosporic ancestor give to the heterosporic ancestor, from where the microspore and consists of microsporangium, where the megasporangium consists of megaspores, which is later on developed into a seed habit. For example, most of the species in Selaginella are heterosporous. Only one functional megaspore mother cells will rise to the 4 megaspore after meiosis. Only a single functional megaspore in a sporangia is present in the Selaginella rupestris, Selaginella monosperma, and Selaginella erythropus, like spermatocytes. Development of female gametophyte, that is endosporic. Fertilization after embryo development take place within the megasporangium. The seeds developed into the pterophytes can be called as a true seeds because cannot be called as a true seeds because they are not covered with the megasporangium are not covered with the integuments, and there is no resting period after the embryo development, and the development of embryo is accomplished within the development of shoot and the rhizopore. So these are the important things of the seed origin. Thank you.